Great. Looks like we're recording. All right. Well, I'm really pleased to introduce Dr. Paulo Campregher from Albert Einstein University Hospital in Sao Paolo, Brazil. He's going to speak to us today a little bit about using RNAseq for gene fusion detection. So without further ado, I'll hand it off to you, Paulo. I know that you said that some of your team were joining us today as well. So I'm looking forward to learning from you. Thank you.
Yeah. All right, Beth. So thanks for the invitation. It's a pleasure talking to you today. I'm going to start sharing my screen. So host disable participant screen sharing. You have to enable me, Beth.
That should do it. Let me know if that worked. Sorry about that.
That's okay, no problem. Right, can you see my screen?
Yeah.
Okay. So today we're going to talk about RNA analysis for identification of gene fusions in cancer. I'm going to talk a little bit about some basic concepts, some challenges, and directions the field is moving, compare a little bit with DNA-based methods, and show some of our cases that illustrates very well how this is useful. For this talk, my disclosures are that I have received the speaker fees from Illumina. So let's start with gene fusions in cancer, right? So why to study gene fusions in cancer? Several very important reasons. So first, we have many oncogenic gene fusions that contribute to causing cancer. We find relevant gene fusions in multiple cancer types. The most important ones, I would say, would be the hematological malignancies, both lymphoid and myeloid malignancies. This is also very important in sarcomas, in central nervous system tumors, and also in several types of solid tumors, typical solid tumors such as lung cancer, prostate cancer, and etc. So gene fusions are very heterogeneous. That means that they don't look always the same. They are kind of tricky to find and to interpret, to curate. They're highly relevant, so many times they are the genomic event that will enable us to make a specific diagnosis, such as BCR-ABL, many other sarcomas and CNS tumors too. They are very important for prognostic reasons, so mainly in the field of hematological malignancies. Some fusions are associated with favorable prognosis, such as RUNX1, RUNX21 fusion in AML that is associated with a better diagnosis. Others are associated with a very dismal prognosis, such as DEC-CAN fusion in AML2. And many times they are therapy targets. So we have several drugs that will target specific gene fusions, so the patient will have very important clinical response to specific therapies. They are very assay dependent. Our ability to detect the fusions are assay dependent. So amplicon versus capture, when we talk about library prep, we also have DNA versus RNA, and so on and so forth. And we have challenges in the curation of fusions. No guidelines yet. Soon we will have a guideline for interpreting gene fusions in cancer. So starting with a definition, a good definition is that a gene fusion is a gene formed by the union of portions of at least 2 different genes. We have some cases of 3 genes involved in fusions in the literature, but we need at least 2. And how are gene fusions formed, right? So we have 4 main events leading to gene fusions. So we have gene deletions, or better saying, chromosomal deletions, right? This is an example where this region is deleted, and that will lead to the juxtaposition of two genes, leading to a gene fusion. We have gene duplications. This is an internal tandem duplication that will lead to the fusion KAA 1549 BRAF. So a segment involving these two genes is duplicated, and that lead to the gene fusion. We have inversions, chromosomal inversions. The typical example, EML4-ALK. We also have inversion 16 in leukemia, and many other examples. And translocations, chromosomal translocations, where we have a piece of one chromosome stick together to a piece of other chromosome, just opposing two genes, such as we see in BCR-ABL fusion with the Philadelphia chromosome. And another relevant point for us to pay attention is where are the breakpoints located, right? The breakpoints could be inside two genes, or one breakpoint could be in an intergenic region and inside a gene, right? When we are always talking here about two breakpoints, or both breakpoints could be in intergenic regions. And this will lead to fusions with different flavors, let's call that, right? Some fusions will produce a chimeric mRNA. Other fusions will not produce a chimeric RNA and will exert its pathogenic effects in other ways. So we can divide fusions in two big groups in cancer, right? So in one fusion, we have two genes, the coding sequence of two genes, or not exactly the coding sequence, but the mRNA sequence of two genes put together. That will lead to a chimeric transcript, and that will lead to a chimeric protein. And in other situations, we have an enhancer of a gene juxtaposed to another gene, and that will lead to overexpression of a normal protein, right? So in this case, we don't have a chimeric transcript. We have, when we do RNA sequencing, we find just a normal transcript. But when we talk about normal proteins, I mean, when we talk about overexpression, let's say this mechanism, right? So when we don't have a chimeric protein, we still have two different flavors for this situation. So in one situation, we have a promoter or an enhancer juxtaposed to another gene, and that will lead to no chimeric transcript. In this case, the transcript we find is just a CRLF2 normal transcript with a very overexpression because it's on the influence of a very strong promoter or enhancer. But we also have another situation where we find a normal protein sequence, but we do have a chimeric RNA transcript. And this is when the breakpoint is in the untranslated region, normally in the 5' UTR, right? So we have this example of a PIK3CA fusion. And in these cases, although we don't have a chimeric protein, we still can detect these products with RNA sequencing. So we still can find this fusion transcript, although we don't have a chimeric protein. And even in this situation, sometimes we can use RNAseq to help us to find this situation, right? So this is a very recent publication where they used, it's a group from Brazil, where they used targeted RNA sequencing with about 13,000 genes. And we can see here that in the cases where we have CRLF2 rearrangement, we can detect most of these cases, in this situation, all of those cases, by the overexpression. So this is not very well validated for clinical use. Validation is kind of tricky with RNAseq, but we can use RNAseq also to identify overexpression and then look for these fusions and find even the fusions where we don't have a chimeric RNA transcript. So gene fusion may consequences, generation of a chimeric transcript, as we just mentioned, promoter swap leading to protein hyperexpression. And we also have a third kind of fusion that will lead to gene inactivation, right? So if we break like a tumor suppressor gene in the middle of the gene, that can be a mechanism of loss of function. This is not very well standardized how to deal with that. I mean, I think we have to move forward in the field with how to interpret when we find gene fusions involving tumor suppressors, and many times they are out of frame transcript. So we still have a way to go here, but this is a well-documented event, right? So talking a little bit about methods, right? So basically what we have here is breakpoint in two genes that will be joined. In this case, I'm showing a chimeric transcript and leading to a chimeric protein. If we're looking at breakpoints, then we have to study DNA, right? Most of the breakpoints occur in introns. We have to sequence DNA in order to find these fusions. If we look into the chimeric transcript, the best approach is no doubt to study RNA. And protein, we have very few ways of looking at fusions, studying protein. We have some immunohistochemistry assays, such as ALK immunohistochemistry, that is a good surrogate for gene fusions in lung cancer, but it does not detect the fusion itself. It detects the hyperexpression of a part of the protein, right? So DNA analysis, we're talking about FISH, PCR, whole genome sequencing, or target hybrid capture. When we talk about RNA analysis, RT-PCR, nanostring, whole transcriptome sequencing, targeted hybrid capture or amplicon-based assays, and protein immunohistochemistry, Western blot, and flow cytometry, right? So that will be a good way to look. Every time we use DNA, we need to study with hybrid capture, right? There's no good amplicon-based, DNA-based assay to detect fusions. But with RNA, we can use amplicon-based assays, hybrid capture, or hybrid capture will be both whole exome-like or smaller panels, or mRNA enrichment assays. When you look at the literature, looking for comparisons between different methods for detection of gene fusions, we don't have a lot of papers. This is a good one, published 2 years ago, where they compared in parallel 5 different approaches for gene fusion detections. So one DNA-based with hybrid capture was an Agilent panel. 3, one RNA amplicon. This is the ArcherDX. The chemistry is a little different, right? Because they use adapters so they can detect fusions with unknown partners. It's an RNA-based assay. 2 and 3 others. So 2 amplicon-based and 1 capture-based. I'm not going to go into all the details, but basically the only approach that captured 100% of the control fusions they had was hybrid capture RNA targeted assay, right? So the other panels had either a problem with sensitivity, so they would not detect lower levels of the fusion, or the fusion studied would not be in the panel. And the only one with all the green, meaning that all fusions were detected, was RNA-based assay with hybrid capture. And we also had this publication in the last ASCO from 2022. It's an abstract from Tempus, where they compared a big number of samples, where they did a hybrid capture DNA-based assay for fusion detection, and in the same sample, an RNA-based assay with hybrid capture too. And what they found was that many gene fusions were detected only in the RNA assay, meaning that many fusions were not detected in the DNA-based hybrid capture assay. And we see that in clinical practice, right? We see many times, we had several cases here in our hospital where a patient did a DNA-based assay, and the assay was repeated using that RNA-based assay, and we did find the fusion. So they found 23% more actionable fusions studying RNA than studying DNA, right? Some genes call the attention, right? BRAF, they had a big number of fusions detected in the RNA only, and the samples were 76 samples. So according to current evidence, RNA analysis is more sensitive for gene fusion detection than DNA analysis. So the best nucleic acid to study is RNA when we're interested in gene fusion. And studying RNA, we have other advantages. So other than the fusions, we can find splicing alterations by itself, right? Different than finding the splicing mutation in the DNA, you can see the splice event by itself. We also have the potential to explore gene expression profiling for diagnosis and hyperexpression or underexpression of genes. But when we look at the total RNA in a cell, we see that the messenger RNA, that is the one we're more interested in, is a very small amount of the total RNA we have in cells, right? We have a lot of ribosomal RNA that we are most of the time not interested in, transporter RNA and other long no coding RNAs and other types of RNAs. So we need some methods for enrichment for either messenger RNA or specific for genes, right? We have basically three main methods. So one will be poly-A enrichment, right? The RNA messenger, most of the molecules would have a poly-A tail. We can use that for capture. We can do ribosomal RNA depletion, and we can also do targeted capture that will be both a smaller gene panel capture or whole exome RNA capture. And there are not many studies evaluating that, but we have a few. So in this article published in 2017, they studied three different approaches for FFPE samples, evaluating what would be the best option for looking for gene fusions. And actually, in this case, not really gene fusions, they were more interested in what approach would capture mostly exons, right? So they study the protocol called RNAseq. It's an exome-like, so you have hybrid capturing the exons of the genome. Ribo0, that will be a ribosomal RNA depletion method, and TrueSeq mRNA, that will be a poly-A enrichment. And what they showed, and this is FFP samples, right? The kind of sample we mostly work with. The exome-like capture was really the best approach for capturing exons. That's kind of intuitive, right? So we see here, pink is exonic sequences, blue is intronic sequences, and green introgenic sequences. So they extract the RNA with these three different approaches, enrich it with these three different approaches, using samples, both from intact to highly degraded samples, many FFP samples. And we can see here that both for intact or highly degraded RNA, we map a lot more to exons when we hybrid capture than when we do ribosomal depletion or poly-A enrichment. So that's why most people in the field are moving to this approach when studying cancer, right? So you capture all exons you're interested in instead of trying to get rid of ribosomal RNA or enriching in poly-A sequences. And there's another study that compared the same RNA access approach with, in this case, total RNA. And again, as expected, the amount of exonic sequences was hugely increased in this approach, right? And here, just showing in the IGV what happens when you do the total RNA, both with 200 million sequences per sample, 30 million sequences per sample. And this is much more effective. You only need, in this case, 25 million, and you have a really big enrichment in exonic sequences. So targeted capture are preferred for mRNA sequencing, especially for degraded samples, but I would say for non-degraded samples too, right? And when we work in the diagnostic lab, we are always thinking about potential challenges, right? So a gene that kept me a little bit uncomfortable in the recent month was FGFR2, right? Because we work mostly with mRNA-based assay. I mean, we do hybrid capture. And when we look at FGFR fusions are detected in 13% of cases of intrahepatic cholangiocarcinomas, highly promiscuous fusions, with about 140 fusion partners described. And what caught my attention that made me a little uncomfortable is that when we look at the events that are considered oncogenics, about 10% of the cases had what they call FGFR2 rearrangements, right? These approvals had as an assay the Foundation 1 assay, and Foundation 1 assay they use a hybrid capture DNA-based assay, different from Foundation Heme, where they study RNA. And they had 10% of cases with FGFR rearrangements that no partner gene could be identified. And the response rate for the anti-FGFR2 drugs in these samples was similar to the one with fusion detected, right? And we also know recently that FGFR truncating events after intron 17 are oncogenic. So my concern here was that if I could be missing FGFR2 fusions with an RNA-based assay, if I could be missing these 10% of cases, right? So we searched in the literature and we found very, very good article published last year where they really looked into FGFR2 fusions architecture in cholangiocarcinoma. And I was happy to learn that in the 3 cases, they studied RNA for which DNA rearrangement had been detected. The cause of the discrepancy between DNA and RNA assays was an insertion between the breakpoint of FGFR2 and the partner gene that gave the impression of a fusion with a region that could not be met. But those were canonical FGFR2 fusions that were detected in RNA sequence. So what happened? So this is very illustrative, right? So this DNA-based assay, they detected the FGFR2 intron 17. They could not map this sequence anywhere, right? They could see that this was a sequence that did not belong to other gene, but they could not map that sequence. And what caught our attention here is that these are small fragments, right? We don't have a lot of nucleotides here. And what happened in this case was that this was an insertion between FGFR2 and DBP gene. So if we had more sequences here, it will be possible to find the DBP gene here. So when they performed an RNA-based assay in this sample, they could find just a canonical DBP FGFR2 fusion, right? So I was a kind of relief with that, because according to this evidence, when we do an RNA-based assay, probably we will find all these rearrangements, right? They only tested 3, but that's what we have for now. All right, so talking a little bit about bioinformatics, right? And we have several different tools for looking at RNA-seq data and find fusions. And this is an interesting paper published 3 years ago. And they compared 23 different tools for detecting gene fusions in RNA-seq data. And here are they study basically 4 breast cancer cell lines that have been extensively studied. And they took the RNA-seq data and they tried different fusion colors, and to see, they were looking for accuracy and speed, right? How fast these tools can process samples. Here we see all the tools they tested, and here are the fusions in the samples, and here which tool found each fusion, right? And when they looked at this is an interesting graph, because when they looked at median runtime and median accuracy, I would say the 3 top tools were Ariba, StarSeq-R, and Starship. Ariba High-C is only the high confidence called by Ariba. And that was the conclusion. These are the 4 best tools for finding fusions from RNA-seq data. And now I'm going to talk a little bit about our experience with the TrueSight Oncology 500, and also with some other tools. Right, we have the validated for a couple of years now TrueSight Oncology 500. We sequence 525 genes. In the RNA part, we sequence 55 genes, and we look for all these splicing, alterations, fusions, and MSI, TMB, and just the regular SNV, indels, amplifications in the DNA part, right? We modified a little bit the standard fusion call pipeline. So the fusion caller from the Illumina is MANTA. And we also run Ariba, so we use both fusions. And we also have another approach where we found sequences from fusion GDB in the internet, and then we have generated FASTA for all those fusions. So we do both a de novo calling with MANTA and Ariba, and we do a alignment that will be what we call fusion hotspots. So we align our sequences to these known fusion sequences. So we have these actually 3 tools for fusion calling: MANTA, Ariba, and the fusion hotspots, right? And something that is very important for us is the visualization of fusion, right? Ariba has a very nice visualization tool, but we are using Ariba not for long. We're using for close to a year now. So in the past, we want to develop a tool to be able to visualize de novo fusion called by MANTA. So we generate FASTA from the fusions, right? We have a consensus sequence, and we generate a FASTA from these consensus sequences. And then we map our sequences to this FASTA. And for every sample, we have a different FASTA, right? Because if no fusions is called, we don't do that. But if any fusion is called, we generate a FASTA for that fusion, and then we map to this FASTA, and we generate a de novo BAM. At the same time, we do a BLAST X, and we generate a GFF, and then we can look at that at IGV. That has been really helpful, right? So this is an example. We can see this is a EML4 ALK fusion, and this is the BAM generated. We can really see the fusions here, and this was very helpful from our bioinformatics team. When we look at samples, we find fusions in our practice. The tumor types we find most fusions. Well, first we find fusions in about 13% of the cases. We sequence dozens of different tumor types. This is all for clinical practice, right? It's an assay for clinical practice. And fusions are present mostly in prostate, adenocarcinoma, lung, cup, glioblastomas, and sarcomas. This is not a hematological panel, it's only for solid tumors, right? So this is not a comprehensive list, but these are some examples of the canonical fusions we found. And by canonical, I mean the two partners have been described in the same breakpoints for both partners in the literature. So we don't have to spend a lot of time curating these fusions because they are very well-known fusions. So we see here many different tumor types, gliomas, glioblastomas, lung adenocarcinomas, prostate cancer, sarcomas. And here, the actionability of the fusion, right? Sometimes that will enable treatment with some target therapy. Sometimes that will help us establish the diagnosis. Sometimes it even changes the diagnosis a little bit, and so on and so forth, right? And what is interesting is that about half of the fusions we find in our practice are novel fusions, fusions that have never been described. So in these cases, we do spend a lot of time curating these fusions, trying to figure out if this is a oncogenic or likely oncogenic, right? Or, I mean, by default, they are VUSs, right? Because they have never been described. But we want to help oncologists to make sense of their cases. So we do a curation in all those fusions, right? We follow a similar approach of this one described by the paper by Jason Saliba and other members of this group. The approach they follow for N-track fusions. So we look a lot at the fusion structure. If the partner is the 3' partner or the 5' partner, and then we compare with the non-oncogenic fusions. If we expect that partner to be the 3' or the 5'. In the case of kinases, we look if the kinase domain is preserved. We look if the fusion is in frame, and if it's the right orientation, 5' or 3', if the kinase is preserved, and if the fusion is in frame, we describe that fusion in the report, right? We call that this is a variant of unknown significance, but we write a text saying to the oncologist that we believe that fusion is probably oncogenic. And now I'm going to show just 3 examples of how this novel, this is a novel fusion, the other one is not a novel fusion, but how we can use this assay. So case 1 is a 16-year-old male, a patient that had a diagnosis of acute myeloid leukemia with translocation 6/9, DEC 214. It's a high-risk fusion. So, and the patient had also eosinophilia when he presented with AML. He had at diagnosis a FISH performed for PDGFR alpha, beta, and FGFR1. For some reason, there was a false negative FISH. And the patient was treated with a hematopoietic stem cell transplant in first complete remission. He had a first relapse around the day 100 after transplant. He was treated with infusion of donor lymphocytes and chemotherapy. And then he had a second relapse, right? Very dismal prognosis, second relapse after transplant. It's a very difficult to treat situation. And here we had a comprehensive genomic profiling, RNA-based, and we could identify a novel gene fusion, never described before, TBL1XR1 with PDGFR beta. That's why I say that the Fisher diagnosis for PDGFR beta was a false negative result. When you look at we published this case in a letter that was in 2017. When we look at the anatomy of the fusions, we see that here we have PDGFR beta, TBL1XR1, and this is the fusion, right? So the fusion was with exon 7 of TBL1XR1 with exon 12 of PDGFR beta, tyrosine kinase domain totally preserved, and the fusion was in frame. When we compare with other fusions described in the literature, we see that exon 12 is a very common breakpoint, right? Exon 11 is another common breakpoint. So exon 11 and 12 are we could call canonical breakpoints. So the oncologist, although there was a novel fusion, he considered that for treatment, and patient was treated with dasatinib, that is a tyrosine kinase inhibitor that targets, among other genes, PDGFR beta. Eosinophilia resolved shortly after initiation of dasatinib, and then the patient was treated with chemo, radiation, dasatinib, and survived for about 36 months. So he had unfortunately, after 36 months, he had a CNS, central nervous system relapse, and passed away of acute myeloid leukemia. But at least this patient gained 3 years after a second transplant, a second relapse after transplant. This is not usual. And so this was very helpful for the patient. I want to present a second case. This was a newborn that presented with this complete blood count in the first day of life: 58,000 leukocytes, 72,000 platelets, hemoglobin of 14. The team performed a bone marrow smear with 60% of blasts. So the diagnosis was congenital acute myeloid leukemia, and he had what is called a blueberry rash, right? So he was born with this skin rash that is typical for some diseases, including acute myeloid leukemia. Skin biopsy also conferred leukemia cutis, leukemia infiltration in the skin. When we look at congenital AML, it's a very rare disease. That is acute leukemia diagnosed before 28 days of life, very rare. It's frequent to find hepatosplenomegaly, monocytic differentiation, and leukemia cutis. These three features the patient had. And the most common genomic finds are KMT2A fusions, translocation 122 and translocation 816. 3-year survival varies from 25 to 35%. There are some reports of spontaneous remissions. And then we perform whole transcriptome sequencing in a research project for this patient. And we could find translocation 822. The patient had a gene fusion involving CAT6A with EP300, right? And this is the output of ARIBA. It's very useful output because it tells us exactly in a visual way where the breakpoint is, what exons from one gene and what exons from other genes are preserved, and also the domains, right? The domains from both proteins that are preserved. I want to call your attention for the very low number of reads here, right? We working with targeted RNA assays in the literature, on our practice, with a small set of genes, we normally see dozens or hundreds of supporting reads. So this was a whole transcriptome assay, and we only saw 3 split reads and 1 discordant mate, right? So this is something we see when we compare targeted assays with whole exome-based RNA assays, right? So when we look at this fusion, we only find 4 descriptions in the Mittelman database of this same fusion. But they have been described in congenital acute myeloid leukemia. And we have this publication from 2018 describing this same fusion. MOS is another name for KAT6A, and the patient had a spontaneous molecular remission. I mean, AML was solved by itself. Unfortunately, our patient the clinical condition was deteriorating, and he was treated with induction and consolidation therapy, and is in remission 1 year after diagnosis, and hopefully it will continue in remission. And now, just to finish, I want to show just one more case of a patient with lung adenocarcinoma, and this is a case we looked into. We evaluated using the TruSight Oncology 500 in our clinical practice. So it was a male with 53-year-old male with lung adenocarcinoma, and this patient had two findings in the DNA, right? One deletion in the MET gene and one NF1 mutation. We see here is a 24 nucleotide deletion, and it's exactly here on the border of exon 14 with intron 13. Right? It only deletes the first nucleotide of the exon, but it deletes the canonical splice site, right? So exon 14, first hypothesis is that this is a exon 14 skipping event. But when we look at the DNA-based assays, this is a Foundation Medicine publication from 2016, cases with MET exon 14 skipping. In the DNA, this is a very heterogeneous event, right? So each one of these small, it's one patient, right? So we have hundreds of different DNA-based events leading to MET exon 14 skipping. So it's a little tricky to interpret this find when you find on an RNA base, but our pipeline called the fusion, the MET exon 14 skipping. And when we look at the IGV, it's very easy to spot the event, right? So these are 2 control cases. So this is exon 13, exon 14, and exon 15. We see no coverage for exon 14, and pretty much all sequences here are from exon 13 to exon 15. So we could, right? Sorry, the report is in Portuguese, but we could state that this patient had a skipping of exon 14 of MET. So this is just to illustrate how RNA-based assays can help us also in splicing events. And with that, I want to finish. I have many people to acknowledge, all these people working with us in the lab and the diagnostic lab, also my other colleagues from clinical practice that help us doing this assay, and also Elvis and Flavia from University of Ribeirão Preto, that we had a partnership in one of the cases I presented here for you today. The patient was their patients, and they sent a sample for us to sequence. And we can have questions if you guys want. Thank you very much.
Thank you.
Dr. Paulo, you have 2 questions at the chat. They're asking you about loss of function fusions and also about long read. So about long read, if you want to answer.
Yeah, well,
important.
You know, I had some long read slides, but I thought it was going to be too big, the presentation. I mean, long read sequencing surely is it's a very interesting approach for gene fusion detections, right? The problem we had with long read was the low quality of bases. That it's getting better, but when you're dealing with gene fusions, the specific quality of each nucleotide is not that relevant, right? The most important thing is to map your event to a fusion gene. So surely it's a very promising approach. We are planning to explore that in the coming months, to evaluate gene fusions using long-read RNA. And it's interesting because you can have the results also in real time, right? You don't have to wait for the experiment to finish. As soon as you have a critical number of fusions detected, you can pretty much say you have found a fusion. And that can be useful for clinical practices. For instance, one problem we have nowadays in hematology is that we have some drugs like anti-CD33 monoclonal antibodies that should be used during induction therapy. And in most of the cases, we only find low-risk fusions that are the situations where these drugs are more useful after induction is finished. So we need to improve our turnaround time, and maybe long-read RNA will be a better way of doing that, right? And about loss of function, let's see in the chat here. Yeah, I mentioned that briefly, SORA. We don't know exactly how to do that, right? When we find a transcript in an RNA-based assay, like with a tumor suppressor gene involving either TP53 or BRCA, can we call that a loss of function event? I'm not sure if we can do that, right? Most of these loss of function events in tumor suppressors have been standardized in DNA-based assays. So I think this is an open question, but it's very relevant. We spend a lot of time thinking about that. I think Andrea has a question.
Yes, thank you. I hope you can hear me. Great presentation. I have a question, I think related to what also Brandon is mentioning in the chat. And I was kind of surprised, well, not surprised, but definitely, the example that you had using RNAseq with so, few reads, but you're still able to call it. Can you comment on maybe the practice, what's your best practice that you use when you have perhaps, targeted assay with, more support versus, more general, like whole transcriptome with, the support might be very few, and, heterogeneity of tumor tissue perhaps that's, would dilute a little bit the signal. And of course, for RNA, whole transcriptome, we know that old methods tend to be oversensitive. And if you can that would be great.
That's a great question, Andrea. And it's these are challenges we face every day. So the transcriptome was a research project, right? So that was not our practice. But answering your question about how do we interpret and what are the cutoffs, right? I think that's basically the question, right? So I think there are a lot of things to talk about. So first, if we talk about a cutoff based on our validation, we need at least 20 reads, including split reads and spanning reads to support a gene fusion, right? That would be the hard cutoff, I would say. But we have several situations that are very, very challenging, right? So for instance, we receive samples that have not been optimally processed, and the quality of DNA and RNA is not optimal. And sometimes we find a few reads in samples like that are very relevant for the tumor type we are studying. So we have recently launched a suspected fusion in the report, right? And then we explain why it is suspected, right? We had a very recent example of that. There was a child with pilocytic astrocytoma, and we did the assay, and we find KAA 1549 BRAF fusion. That is the canonical event in this tumor type. But with a it's less than 20 reads. I think we probably have 5 or 10 reads, and both split and spanning reads. So we did that, and there was a sample process outside our hospital. The quality was not optimal. So we wrote that, we found a small number of reads of Ka 1549. We kind of give that information to the oncologist for him to deal with. If we find like out of frame fusions or any other kind of event in the same situation, that's not going to the report, right? So we take a lot of that into consideration, right? So this looks like an oncogenic fusion. No, if it doesn't look like an oncogenic fusion, we have 4 or 5 reads, we don't talk anything about that. If it really looks like an oncogenic fusion, but the reads are not optimal, we somehow give that information to the clinician, stating that the quality control was not optimal, we had problems, and then he can decide if he makes another biopsy for another sample. So we take all of that into consideration. But our hardcore, cutoff will be 20 reads, having both spanning reads, split reads, and one more point. We also need to have that stair-like mapping, right? Because sometimes we have a single block. Just like as if it was an amplicon-based assay. If that's the case, small number of reads, single block, we also don't consider that robust. So when we look at IGV, we need to see that we have different starting point and ending points to consider that a sound oncogenic, a sound fusion, right?
Great, thanks.
Yeah, it's kind of complex. There's another question. Have you folks encountered any issues with RNA-based approach over calling fusions being too sensitive? One colleague who works in fusion calling for pediatric patients with cancer has looked at multicolor approaches. Yes. So when we approached that on our validation, right? So we didn't find an excess of fusions called when we compare with other samples. And we also are CAP accredited. So every 3 months, we receive samples from College of American Pathologists. And we are doing very well. So with this approach of targeting capture and having this cutoff of 20 at least with that pattern, we don't find overcall fusions. Of course, we have fusions that don't look real, right? You have 2, 3, 4 reads. And for this very low confidence fusion, we sometimes have what we consider false positive. But if you use the cutoffs, we don't find a big number of fusions. We don't believe they're fusions.
That's great. Thank you.
So kind of to piggyback on that idea, do you ever rescue those low confidence fusions that are maybe just 3 or 4 reads back into possibly we'll report this if we can confirm it, if it's something that might be diagnostic for the patient?
Yeah, if it's important, we don't have a mechanisms for validation, for validating that by ourselves in the lab, right? This is a diagnostic lab. We have 2 weeks turnaround time. We have to decide what to report or not, right? It should be reported. But that decision, many times we talk to the oncologist or hematologist, right? And then we explain the complexity of the situation. And then we kind of decide together if that patient needs another type of confirmation with a different assay. And then we can do that. For instance, we had a case recently also that we found an ALK fusion on a sarcoma with a small amount of reads. And we know that as a rule, there is a very good correlation with ALK fusions and immunohistochemistry, right? Most ALK fusions, they do show hyperexpression in immunohistochemistry. So we suggested, we talked to the oncologist, let's do a immunohistochemistry. So That's the other way around, right? Normally, we do immunohistochemistry first and then genomic assay to confirm. But in this case, we decided to do the opposite. And we performed the immunohistochemistry, and that was negative. So now it's up to the oncologist to decide if he considers that a useful target. You know, the fusion was there. I mean, we could say that, but the breakpoint was not canonical. And then we went to study ALK fusions in sarcomas. They have weird breakpoints also when we compare with lymphomas or lung adenocarcinomas. In sarcomas, the breakpoints are not canonical ones. But yeah, sometimes we have to try to talk to the oncologists. And I saw you mentioned you often get low numbers, right, for BRAF2. Yeah, that's a problem.
It's just a tricky fusion altogether. So, yeah. So yeah, it's good to know that we share the same challenges.
Yeah.
Well, we're at the top of the hour. We don't have any more questions. I just want to thank you so much for coming here today on this forum and presenting on RNAseq. I know I definitely learned a lot from you, and we had a really great turnout today. So thank you so much for sharing with us, and everybody have a wonderful day.
Thank you, Beth. It was a pleasure. Bye-bye, guys.
Bye.